أهلا بكم أبنائنا وبناتنا طلاب وطالبات الثانوية العامة في ريفيجن على مادة البيولوجي. احنا النهارده الحقيقة هنراجع كل البيولوجي في شكل scientific terms. حل scientific terms مهم جدا لأن دي الكونسبتس بتاعة المنهج. مش بالضرورة تبقى السؤال ده يجي scientific term بس، لا أنا بعرف المعلومة ممكن تيجي multiple choice، ممكن تيجي recorrect، ممكن what's meant by، ممكن هتيجي في اي شكل من اشكال الاسئله، لكن المهم ان احنا نعرف المعلومه. هنراجع المنهج كله ان شاء الله في خلال ساعه واحده بشكل سريع جدا لان احنا بنعمل ريفيجن، فيدوب هنبدا نحاول نغطي كل الكونسبتس بتاعتنا. هنبدا باول حاجه كده. Give scientific term for each of the following. Cavity fits the head of thigh bone. Cavity fits the head of thigh bone or the femur bone. It is the acetipulum cavity. So the acetipulum cavity is the cavity where the head of the femur fit inside it. The next question. A rounded bone lies anterior to the knee joint. The question asks about a round bone on the knee joint. It is the patella, and also it's called the kneecap. The next question. A triangle bone with an anterior head end and pointed outer one. What about the triangle bone in the human skeleton? It is the scapula. The scapula is the triangle bone. The inner prone end is pointed where the presence of the cavity, which is called the glenoid cavity, where the head of the humerus fit inside it. The next. A flat bone pointed at its lower parts, which made of cartilage that act as a bone in the thoracic cage. It is called the sternum bone, where the lower part of the sternum is made of cartilage. The next. A curved bone attached to the vertebra at its centrum and its transverse process. The curved bones that attach to the vertebra at two sides. The first is the centrum of the vertebra. The second is the transverse process. Of course, that is the rib. 12 pairs of ribs form the rib cage in addition of the sternum bone and the 12 vertebrae. The next. A girdle consists of two identical halves of scapula and the clavicle bone. Of course, this girdle is the shoulder girdle or pectoral girdle. The next. A joint consists of two rows of 8 carpels. Two rows of 8 carpels form the joint, which is called the wrist joint. The 8 bones are formed in two rows that form the wrist joint. The next. A girdle consists of two halves fused at the pubic symphysis. The girdle consists of two halves fused at the pubic symphysis. It is the pelvic girdle, where the pubis of each half is connected anteriorly or at the ventral side to form the pubic symphysis. The next. A deep depression at the connection between the ileum and the ischium. The depression or the cavity between the ileum and the ischium. Of course, that is the acetipulum cavity where the head of the femur fit to form the thigh joint. The next. Part of the hind limbs supported by two bones, inner tibia and outer fibula. It's of course that the shank bone, which consists of the inner big tibia and the outer small fibula. The largest bone of 7 tarsals that form the ankle. Of course, the first or the largest bone in the ankle is the heel bone. The ankle consists of 7 bones which are called tarsals, but the rest consists of 8 bones which are called carpels. The next, the movement of cytoplasm inside the living cell. The cytoplasm move inside the living cell in the circular manner that's called the cytoplasmic streaming. The next, biological expression of movement by tendril and pollen roots that act as a kind of movement in biology by tendril in pea plant, for example, by pollen roots in bulbs. Of course, that is the haplotropism or thigmotropism. The next, the unit structure of the muscular system. The unit structure of the muscular system. The answer is the muscle, because the question asks about the muscular system. The unit, the next, the tissue have the ability to contract and relax, causing a motion. It is the only tissue that can move inside the body, which is called the muscular tissue. So the muscular tissue has the ability to contract and relax, causing a motion. The next, the membrane which surrounds the muscle bundle. The membrane which surrounds the muscle bundle. It's called the premicium. The premicium membrane surrounds a group of muscle fibers are called the muscle bundle. The next, the cytoplasm of the muscle fiber. The cytoplasm of the muscle fiber. The muscle fiber means the muscle cell. So the cytoplasm inside the muscle fiber is called sarcoplasm. So that is the name of the cytoplasm inside the muscle fiber. The next, the membrane which surrounds the muscle fiber. This membrane is called the sarcolemma. So the cytoplasm of the nerve fiber is the cytoplasm is the sarcoplasm, but the membrane is the sarcolemma. The next, a kind of muscles, its myofibrils has dark bands and light bands that act as the striated muscles. So these muscles are the skeletal and cardiac muscles because they are striated muscles, consists of dark bands and light bands. The next, a kind of muscle is striated and involuntary. Striated means containing light bands and dark bands. Involuntary means it's not moved by our will, but it moved by controlling the autonomic nervous system. It is the heart muscles or the cardiac muscles. The next, the distance between each successive two Z lines in the myofibril. The distance between two successive Z lines in the myofibril, it is called the sarcomere. Sarcomere containing one dark band and one light band. The next, a part of the myofibril consists of myosin only. This part which containing myosin only is the part at the middle or bisect the dark band, which is called the H zone or semi-lighted H zone. The next, a bone that makes semicircular rotation around the fixed ulna. The bone make a semicircular rotation around the fixed ulna. It is the radius, because we know that the radius move in a semicircular manner around the fixed ulna. The next, the semi-lighted that bisect, or the semi-lighted light that bisects the dark band or area that bisects the dark band in the structure of the myofibril. Semi-lighted area, it's act as the H zone because of the presence of the myosin only, and there is no actin in the semi-lighted area. The next, a thick protein filaments in the structure of the myofibril. Of course, that is the myosin, because we remember that the myofibril consists of a group of alternation of dark bands and light bands. The dark band made of thin filaments, the light band made of thin filaments called actin bisected by Z line. But the dark band made of myosin and bisected by semi-lighted area, which is called the H zone, where the presence of the myosin only. So a group of alternation of the light bands and the dark bands together that form the striation of the muscles or the skeletal and the cardiac muscles. The next question. An enzyme which destroy the acetylcholine after performing its function. This enzyme is located at the neurojunction muscles that's called the cholinesterase enzyme. The cholinesterase enzyme destroys the acetylcholine into choline and acetate after performing its function. The next question. A point of connection between terminal branch of the nerve fiber and motor end plate of the muscle fiber. The point of connection between the terminal branch of the nerve fiber means the terminal arborization and the motor end plate of the muscle fiber. This point is called the neurojunction muscle or neuromuscular junction. Neuromuscular junction. The next. The enzyme is located with the enough amount at the neuromuscular junction. The enzyme which hydrolyzes or destroys the acetylcholine into choline and the acetate, it is the cholinesterase enzyme. This is the right answer. The next. A chemical substance accumulated in the muscle causing spasm and fatigue. The chemical substance that causing fatigue in the muscle, that's called the lactic acid. But the lactic acid is formed inside the muscle due to the shortage of oxygen or due to the anaerobic respiration of the muscle during the shortage of oxygen. At the presence of oxygen, the glycogen inside the muscle, it changes into glucose. The glucose is oxidized into pyruvic acid. At the presence of the oxygen, the pyruvic acid is oxidized into acetyl group, can get in Krebs cycle and the electron transport to produce great amount of energy in the form of ATP. But during the shortage of oxygen, pyruvic acid can't oxidize into acetyl group, but in muscles it reduced into lactic acid. The accumulation of the lactic acid in the muscles that leads to the muscle fatigue. But if you still, without a movement, to allow or to get the chance to allow the oxygen to get inside the muscle, that will oxidize the lactic acid into pyruvic acid and they get in Krebs cycle. So that's the formation and the consuming of lactic acid inside the muscle. The next question. A substance that causes the physiological support of the plant. The substance which is responsible for this physiological support in plant is the water. So the presence of water act as a presence of the physiological support. The absence of water means the absence of physiological support. Therefore, the physiological support is called the temporary support. The next. The cell which secrete a fluid to nourish the sperms inside the testes. That's the first scientific term in the reproduction in living organisms, chapter 8. The cell which secrete a fluid to nourish means to feed the sperms inside the testes. The cells are called the Sertoli cells. The next. Cells inside the testes are believed to have an immunization function. Immunization function means they give the sperm the immunity to get a resistance against the infection with any microbes. The cells are called the Sertoli cells. So the Sertoli cells are responsible for the nourish of the sperms and also they have an immunization function. The next. A single cell with a thick coat formed of the nucleus and the little amount of cytoplasm. Single cell with a little amount of cytoplasm and the nucleus, that's called the spore. The next. The ability of unfertilized egg to develop to new individual. The ability of unfertilized egg to develop to produce complete individual, it is called the parthenogenesis, which act as a kind of asexual reproduction. The next. The life cycle of living organism includes 2 generations. One reproduce sexually and the other reproduce asexually. The phenomenon is called the alternation of generation. So the alternation of generation is the Life cycle of the living organism includes two generations. One of them is produced as a result of sexual reproduction, but the other is produced as a result of asexual reproduction. This kind of reproduction to allow the living organism to obtain both advantages of sexual and asexual reproduction. The next question. The transmitters of Plasmodium among human beings. The transmitters of Plasmodium among the human beings means the living organism that is responsible for transferring Plasmodium from the patient to the healthy person. It is the infected female Anopheles mosquito. It is not the female Anopheles mosquito only, but it is the infected female Anopheles mosquito, because the infected female Anopheles mosquito containing the sporozoite which act as the infective stage of the Plasmodium. The next. The infective stage of Plasmodium malaria. Of course, that's a simple question, because the infective stage is the sporozoites, which transfer from the patient to the healthy person by the infected female Anopheles mosquito. So the infective stage of Plasmodium malaria to the human beings is the sporozoites, but the infective stage of Plasmodium malaria to the female Anopheles mosquito is the gametocytes. The next. The infective stage of Plasmodium malaria to mosquito, it is the gametocytes, which is not active inside the human blood, but become active in the cavity of the stomach female Anopheles mosquito. Not active means there is no fusion between male gamete and the female gamete inside the human blood. But when the male gamete and the female gamete, or the gametocytes which are inactive, pass from the human blood or the infected or the patient blood to the stomach cavity, that activates the gametes to be active and fuse together to form the zygote. The zygote change into movable stage, which is called the ookinete. The ookinete can penetrate the wall of the stomach of the female Anopheles mosquito and change into oocyst. Inside the oocyst, the diploid cells 2N divided by meiosis to produce haploid cells inside the oocyst. That takes place by meiosis division. After that, the parasite divided by sporogeny to form a great number of sporozoites, which can migrate from the stomach of the infected female mosquito to the salivary glands of the mosquito to be ready to infect another person. The next question. The human organ where sporozoites spend the incubation period. The human organ where the sporozoites spend the incubation period. Of course, that is the liver, but incubation period, it is the time from the infection and the appearance of the symptoms of the malaria disease or any disease. So the incubation period takes place inside the human liver or the hepatic cells. The next question. The infective stage which produced as a division of merozoites. The infective stage which produced as the division of the merozoites. Of course, this infective stage of the female Anopheles mosquito, which is called the gametocytes. We remember that the merozoites divided by schizogony or fragmentation inside the RBCs, they produce a great number of merozoites to repeat the infection of another RBCs, and some of them change into inactive gametocytes inside the human blood. The next question. The stage that produced from the germination of the polypodium spore. The polypodium spore is haploid. It is produced as a result of meiosis division. But the germination of the spore in mud soil, that will form the gametophyte. The gametophyte is the haploid green heart-shaped plant that containing the antheridia, which act as the male organs that are responsible for the formation of the ciliated sperms, and also containing the archegonia, which is responsible for the formation of female gamete, which is called the egg cell. But the formation of gametes in the case of the gametophyte takes place by mitosis division, but not meiosis division, because the gametophyte is the haploid tissue. And the haploid tissue can't divide by meiosis, but divided by mitosis only. The next question. The male organ of furnace. The male organs of furnace It is the antheridia. The singular of antheridia is the antheridium, that responsible for the formation of the male gamete, which is called the ciliated sperms. The ciliated sperms act as the male gametes surrounded by cilia to swim in water to reach the egg cell for fertilization and the formation of zygote. The next question. The female organ of furnace. The female organs of furnace is the archegonia. The archegonia is the female organ of furnace and divided mitotically to produce the egg cell, which act as a female gamete. The next question. The flowering plants, their seeds develop with a precarp. Precarp, it is the wall which surrounds the fruit. So the flowering plants that are responsible for the formation of fruits and containing seeds, which are called the angiospermia. The angiospermia means the flowering plants which contain covered seeds inside the fruits. The next. A group of flowers on the floral axis in different aggregation. If a group of flowers are located in the floral axis, that's called the inflorescence. But the single flower in the floral axis is called the solitary flower. But inflorescence means a group of flowers on the same floral axis. The next question. 4 cells are produced from meiotic division of the spore mother cell in the anther. The 4 cells are called for microspore because the microspores are haploid cells. They produced as a result of the meiotic division of the spore mother cell. The spore mother cell is diploid, divided by meiosis to produce 4 haploid cells, which are called for microspores. The microspores, each of them, surround itself with the coat, and the nucleus divided by mitosis to produce 2 nuclei. One of them is called the generative nucleus, and the other is called the pollen tube nucleus. The generative nucleus is divided by mitosis to produce 2 male nuclei. 2 male nuclei inside the egg cell, inside the pollen grains. That means double fertilization will take place. Double fertilization means one of the 2 male nuclei is fused with the egg cell to form zygote, and the second will fused with the 2 embryo sac nuclei to form the endosperm 3N. That's called the triple fusion. But the formation of the zygote and the endosperm together, that's called the double fertilization, which takes place by the 2 male nuclei inside the pollen grain. The next question. The transferring of pollen grain from the anther to the stigma of the flower. The transferring of pollen grains from the anther to the stigma of the flower. That's called the pollination. The pollination is divided into 2 types. The first is the self-pollination. The second is a mixed or cross-pollination. If the pollination takes place by transferring the pollen grain from the anther to the stigma of the same flower or another flower on the same plant, that's called self-pollination. But the transferring of the pollen grain from the anther of the flower to the stigma on another flower on the other plant, that's called the cross-pollination or mixed pollination. The next question. A group of cells inside the testes secretes the testosterone hormone. The cells that are responsible for the male hormones or the androgens, that's called the interstitial cells. Interstitial cells is a group of cells between the seminiferous tubules inside the human testes. The interstitial cells at the expression that's located in the cricurium endocytes. The first inside the human testis between the seminiferous tubules. The second is a group of cells responsible for budding in the multicellular organisms at the hydra, which act as a kind of animal. The next question. Tubules inside the testis produce sperms. The tubules inside the testis that produce sperms, that are the seminiferous tubules. The next question. A group of cells inside the testes give an immunization function. That's a very simple question because it is talk about the Sertoli cells, which are responsible for feeding and also responsible for the immunization function or give the sperms and immunity. أصدقائي الأعزاء، فاصل قصير ثم نعود مرة أخرى إن شاء الله. أهلا بكم. نرجع مرة تانية للجروب of scientific terms which act as the lever of the biology curriculum. كنا عملنا ريفيجن على كل شابتر 7 as a scientific terms. قلنا جزء كبير من شابتر 8 وهنكمل دلوقتي مع بعضنا ال concepts of biology. The question asked about the give scientific term. The cells formed from mitosis of testes primary germ cells several times. If the primary germ cells divided several times by mitosis, that produce a great number of spermatogonia. So the spermatogonia are a group of cells formed from the mitosis division of testes primary germ cells, or the primary germ cells inside the human testes. The next question. A stage formed when the spermatogonia store an amount or great amount of food. That talk about the growth stage, where the spermatogonia store great amount of food and change into primary spermatocyte. So the primary spermatocytes are a group of cells which responsible for storing great amount of food inside the spermatogonia. The next question. Stage formed when the primary spermatocyte are divided by meiosis 1. If the primary spermatocyte divided by meiosis 1, that will form secondary spermatocytes. That is the stage of maturation where meiosis division takes place. The next question. A stage formed when the secondary spermatocytes are divided by meiosis 2. That's called the spermatids, which act as the end of the maturation stage. The next, haploid cells change directly into sperms without division. The haploid cells which change into sperms as the spermatids. That is called the metamorphosis. So the spermatogenesis pass through 4 stages. The first stage is called the multiplication, where the primary germ cell inside the testes divided by mitosis many times to produce a great number of spermatogonia. The spermatogonia pass through the growth stage to store great amount of energy inside the cell which divided to produce 4 sperms. So the spermatogonia pass through the stage which is called the growth and they change into the primary spermatocytes. The primary spermatocyte pass through the maturation division, where it pass through the first and the second steps of the meiosis division. The primary spermatocyte pass through the meiosis 1 to change into secondary spermatocytes, haploid cells. The secondary spermatocyte pass through the second meiotic division to produce spermatids. And the spermatids pass through the 4th stage, which is called the metamorphosis, to produce or to change into sperms. So the stages of the spermatogenesis are 4 stages. The first is the multiplication, the second is the growth, the third is the maturation, the fourth is the metamorphosis. But in the case of the formation of female gamete, which is called the oogenesis, pass through these 3 stages only. The multiplication, the growth, and the maturation only, and there is no metamorphosis in the case of the oogenesis. The next question. The phase through which the spermatids are converted into sperms. This stage is called the metamorphosis. Metamorphosis means it changes the shape of the spermatids into the shape of the sperm, which consists of head, neck, the midpiece, and the tail. The next question. An organelle inside the sperm secretes the hyaluronase enzyme. The organelle inside the sperm is the acrosome, which act as a part of the head of the sperm that produce the hyaluronase enzyme, which is responsible for dissolving the hyaluronic acid around the coat, which is around the mature ovum, to facilitate its penetration by sperm. The next question. The cells formed from mitosis of the ovary primary germ cell several times. The question asks about the cells which pass through the multiplication stage of the oogenesis. Of course, these cells which are produced as a result of mitosis division many times is the oogonia. The second question. The periods of placental mammals where the ovary become regularly active in the adult female. The question asks about the periods of the placental mammals where the ovary become regular to produce ova or become active in the adult female, that is the breeding cycle. The next question. The phase when the ovary stimulated the produce or the production of the gravium follicle. This phase is called the proliferation stage. That takes place by the effect of the follicle stimulating hormone which is secreted from the anterior lobe of pituitary gland. The next. The phase when the gravium follicle release the mature ova. That is the second stage which release the mature ova, which is called the ovulation, that to form the mature ova or release the mature ova from the gravium follicle, and the gravial follicle change into corpus luteum. We remember that the pituitary gland or the anterior lobe of pituitary gland secretes the FSH hormone for about 10 days during the stage which is called the proliferation stage. The FSH hormones pass through the blood to reach the ovary and stimulate the ovary to form a gravium follicle. The gravium follicle is responsible for the secretion of estrogen hormone, which is responsible for increasing the thickness of the endometrium. After 10 days of the proliferation, the anterior lobe of pituitary gland secretes the luteinizing hormone or LH hormone. The luteinizing hormone stimulates the gravium follicle to release the mature ova and change into corpus luteum. The corpus luteum produces or secretes the progesterone hormone, which adhere the endometrium inside the uterine muscles and increase its vascularity. If the ovum is fertilized after 1 to 2 days, the corpus luteum will grow to reach the maximum growth at the 3rd month and degenerates at the 4th month of the pregnancy. But if the ovum is not fertilized after 1 to 2 days, the corpus luteum degenerates. The next question. The outer embryonic membrane that surround the embryo and form placenta. The outer membrane that surrounds the embryo is the chorion. The edges of the chorion are connected together to form the placental villi. The next question. A fluid that surrounds the embryo to protect it against shocks and dryness. The fluid around the embryo is the amniotic fluid, which protects the embryo against shocks and also against dryness. The next question. Fusion of two male nuclei, one with the egg cell and the other with the embryo sac nuclei. That's two kinds of fertilization, one of them with the egg cell to form embryo and the other with the two embryo sac nuclei to form endosperms. That's called the double fertilization. The next question. A membrane, its margins fused, forming the umbilical cord. Of course, that is the amnion, which act as the inner membrane which surrounds the embryo, and the edge of the amnion that connected together to form the umbilical cord, which connect between the placenta and the umbilicus of the fetus. The next question. The fusion of male nucleus with the two embryo sac nuclei to form endosperm, that is the triple fusion. Triple fusion because 3 nuclei are fused together. One of them is the male nucleus and the 2 embryo sac nuclei to form 3N endosperm. The next question. The exchange of pieces between internal chromatids after pairing of the homologous chromosomes. That takes place in prophase 1, in meiosis 1, during the meiosis division that called the crossing over. So the crossing over is a change or exchange between the inner piece of the chromatids at a certain point, which is called the chiasma. The next question. Nutritional tissue surrounds the embryo sac inside the flower ovary. The nutritional tissue surround the embryo sac inside the flower tissue. Of course, that's called the nucellus. The nucellus is the tissue surrounding the embryo sac to feed the embryo sac and supply the embryo sac with a suitable amount of food. The next question. The flower cells, which result from the meiotic division of the spore mother cell during the pollen grain formation. The four cells which result from the meiotic division of the spore mother cells during the pollen grain formation. Of course, 4 microspores. The next question. The fruit in which the receptacle or any parts sharing the ovary to store food, as in apple. This fruit is called the false fruit. False fruit is a fruit where any part of the flower sharing the ovary to form the fruit, that's called the false fruit, as that in apple. The next. The ability of the ovary to develop into seedless fruit without pollination. Of course, that's called the parthenocarpy. We should remember that the ability of the animal ovum to develop to produce complete individual without fertilization, that's called the parthenogenesis, which act as a kind of asexual reproduction. But the formation of seedless fruits or the stimulation of the ovary to change into fruit without seeds, that's not act as a kind of reproduction, but just the growth of the ovary and the change into fruit. The next question. The ability of the ovary to develop into seedless fruit by using auxin. That's act as a kind of parthenocarpy, but by the interference of the human beings, so that's called the artificial parthenocarpy. The next question. It's a branch of biology which studies the transmission of characteristics from the parents to the offspring generation after generation. That's the first question in the genetics. So this branch of biology is genetics. The next question. The value and the benefit of any experiment performed is determined by the degree and the adaptation of the tools and the materials to the experiment. That's the expression of Gregor Mendel, which is called the adaptability. So the adaptability is the value and the benefit of any experiment performed is determined by the degree of the adaptation of tools and materials to the experiment. The next question. When two homozygous individuals are different in a pair of allelomorphic characters are crossed, then the dominant character appear in the F1 or the first filial generation, while the two characters appear in the F2 or second filial generation in the ratio of 3 dominant to 1 recessive. That's the Mendel first law or law of segregation. The next. It is the description of the genes which the individual contain. That means that's the genotype, which is the description of the genes which the individual contains, means it's pure or impure. So the answer is the genotype. The next question. An organism carries 2 similar genes for the character. The organism which carry the 2 similar genes of the character, that's called the homozygous or pure individual. But 2 different genes, that's called the heterozygous. Organism carry 2 different genes for the character is the heterozygous. But 2 similar genes of the character, that's called the homozygous. The next. The opposite character with the clear difference. The opposite characters with the clear difference. That's called the allelomorphic character. The next. When 2 homozygous individuals are different in 2 pairs of allelomorphic characters are crossed, each pair is assorted at random and is inherited independently of the other one, and will appear in the second filial generation in the ratio of 3 to 1. That's the Mendel second law or law of independent assortment of genes. Law of independent assortment gene. The next. Geometrical drawing like chess show the male and the female gamete. That is the drawing of the Punnett square. The Punnett square show the male and the female gamete and the offspring is produced as a result of the crossing. The next question. The building unit of the nucleic acids. That the first scientific term of the molecular biology. The building unit of the nucleic acid is the nucleotide. Nucleotide of DNA is called the deoxyribonucleotide, but the nucleotide of RNA is called ribonucleotide. The next. Testing the dominant character if it's pure or impure by crossing the individual which is with the recessive one. That's act as a question of the genetics. Testing the dominant individual to describe if the individual is pure or impure. That's called the test cross. Test cross is a test of the dominant individual to indicate if it's pure or impure. But there is no test cross in the case of the lack of dominance, because in lack of dominance each phenotype has its own genotype, or each genotype has its own phenotype. In the lack of dominance, 3 phenotypes with the 3 genotypes. So the impure individual carry an intermediate character, therefore it doesn't need to carry out test cross. And also in the 6-linked character of the human Drosophila and the mammals males, because the males are pure because it has 1 X chromosome, carry 1 gene only. It's may be pure, dominant or recessive. So the individual in the 6-linked character in the XY structure is pure. Next. An inherited case that's controlled by 2 genes, but no gene is dominant over the other, but they have equal strength and produce an intermediate character. Of course, this case is a lack of dominance because there is no gene dominant over the other one, and no small letters is used in the genetic problems of the lack of dominance. The next. An inherited case that's controlled by 2 pairs of genes. The dominant character appear only if the 2 dominant different genes are present in pure or hybrid state, where each one complete the function of the other one, and the F2 will be 9 dominant to 7 recessive. That's the complementary genes. In the case of complementary genes, the dominant character appear only at the presence of 2 dominant different genes. The next question. Genes leads to stop the growth and death of a quarter of the members in different life stages if they are found in pure state. That's called the lethal gene. The lethal gene is a gene that leads to death due to the stop of the functions that are responsible for the continuance of the life of, in the case of the pure individuals. The next question. The character which appear in the ratio of a quarter of the individual of the F1, that's in the Mendelian cases. These character which appear in the ratio of quarter is the recessive character, because the recessive character is not appear in the F1, but appear in the F2 with the ratio of 25%. Because Mendel state in the Mendel first law, when the 2 homozygous individuals are different in a pair of allelomorphic characters are crossed, the dominant character only appear at the presence at the F1, while the 2 characters appear in the F2 in the ratio of 3 dominant to 1 recessive. The next question. Human genes that located on the chromosome number 8. That question talk about the fingerprint, which act as one of the genes that located on the chromosome number 8. The next question. An inherited case that's controlled by more than 1 pair of allelomorphic genes that present on the same position on homologous chromosomes, but each individual carry only 2 of these alleles, as that of the example of the human blood groups. this case is called the multiple alleles. Multiple alleles means there is more than one pair of genes. Each individual has one pair only, and the three genes in the case of the blood groups A, B, and O, each individual carry two genes only. We remember that A is dominant over O, B is dominant over O. Between A and B, lack of dominance. Therefore, the three genes of the blood groups A, B, and O produce four phenotypes. The four phenotypes are A, B, AB, and O. But the four phenotypes are produced from the six genotypes. The six genotypes means A has two genotypes, AA, which is the pure case, AO is the impure, and also B has two genotypes, BB, which is the pure, and PO is the impure. But the AB, which act as the intermediate character because of the lack of dominance between A and B, and O is the recessive character, which is the genotype is the OO. Therefore, in the case of the blood group, three genes produce four phenotypes and six genotypes. The next question. The blood group which has no antigens and can transfer safely to any other blood groups, that is the blood group O negative, because O negative act as a universal donor, because the O negative has no any antigens on the surface of the RBCs. The next question. The blood group which has no antibodies and can receive safely any other blood groups that act as a universal recipient, which is the AB negative blood group. The AB blood group has no antibodies, but the AB positive, or positive means containing RH, it can to produce the antibodies against A, B, or RH. But the AB negative can produce antibodies against RH if transferring of blood takes place for one time only. The next question. Antigen found in about 85% of the human beings and discovered in the blood of the same monks, some monks, before it was discovered in the human blood, and controlled by three pairs of genes which are carried on the same chromosome. This antigen is not A or B, but the rhesus antigen or RH factor. The next question. Genes carry on the same chromosome and transfer from the parents to their offspring as one unit. These genes are linked on the same chromosome, therefore that's called the complete linkage. The next. Genes are not linked completely due to the crossing over, but they are inherited independently. The crossing over takes place to break the complete linkage and change it into incomplete linkage. The next question. The exchange of pieces between two inner chromatids of homologous chromosomes in the prophase 1 of the first meiotic division. The exchange of pieces between the inner chromatids is called the crossing over, which takes place in meiosis 1, also prophase 1, which act as the first stage of meiosis. The next. A diagrammatic drawing determines the location of genes on the chromosomes. It depends on the result of linkage and crossing over that takes place by Morgan, which is called the genetic map. Genetic map is a description of the genes that link on the same chromosome or determines the distance between the genes which are located on the same chromosome. A modern science explains the study of the molecular basis of inheritance. The molecular basis of inheritance is the DNA, so the branch of science that's called the molecular biology. The next question. The breeders of the genetic information of any living organisms. The genetic information carried on the chromosomes and the chromosomes located inside the nucleus. So the genes which act as a genetic information or the DNA is located on the chromosomes of the eukaryotic living organisms. The next. A phenomenon in which some of the genetic materials form from a heated virulent bacteria had entered in the non-virulent ones, transforming them to the virulent bacteria, or the phenomena in which the non-virulent bacteria change into virulent bacteria by using the genetic materials of virulent bacteria. That's called the bacterial transformation. Bacterial transformation means transforming the non-virulent bacteria R into virulent bacteria S by using the genetic material of the virulent bacteria. The next. An enzyme hydrolysis DNA completely doesn't affect the protein or RNA. The enzyme which hydrolyzes DNA is called the deoxyribonuclease enzyme. We explained the deoxyribonuclease enzyme in the crucial experiment, which proves that the DNA is the genetic material. When the isolated material of the virulent bacteria by Every is treated with the deoxyribonuclease enzyme, the bacterial transformation stops. That means the bacterial transformation takes place by using the genetic material of virulent bacteria. But the isolated material by Every containing DNA and the impurities of protein. If the deoxyribonuclease enzyme causing a complete hydrolysis of DNA, that means the isolated material containing protein or RNA but not containing DNA. So the bacterial transformation stops. That means there is no genetic material in the sample. The next question. A part of DNA strand show the alternation between the phosphate and sugar groups. That's called the sugar phosphate backbone of the DNA. The next. A material remains stable in the cell of the living organisms. The material is the genetic material, which is the DNA. A chemical bond that explains that the combination between the nitrogen spaces and the carbon atom of the deoxyribose sugar. The chemical bonds are the covalent bonds that connect with the other structure with the sugar. The next. A building unit of DNA molecule. It is the deoxyribonucleotide or nucleotide. The next. A group of nitrogen spaces has single ring. That's called the purine nitrogen spaces as A and G. A and G is a purine nitrogen spaces made of double rings, but the pyrimidine nitrogen spaces made of single ring as a T and C, or thymine and cytosine. So the nitrogen spaces are divided into purine double rings and pyrimidine single ring. My dear students, that's the end of this episode. Thank you very much. Goodbye and good luck.